Hello, my name is Sergio Ibarra, and today I'll be giving a summary on Chapter 3, Bioenergetics of Exercise and Training. The objectives for today will be to be able to explain the basic energy systems available to supply ATP during exercise, understand lactate accumulation, metabolic acidosis, and cellular manifestations of fatigue, identify patterns of substrate depletion and repletion during various exercise intensities, describe the bioenergetic factors that limit exercise performance, develop training programs that demonstrate metabolic specificity of training, and explain the metabolic demands of and recovery from interval training, high-intensity interval training, and combination training to optimize work-to-rest ratios. The key terms for this chapter include bioenergetics, catabolism, anabolism, exergonic reactions, endergonic reactions, metabolism, adenosine triphosphate, otherwise known as ATP. We'll start off with chemical structure of an ATP molecule. So an ATP molecule contains adenosine, which is adenine and ribose, the triphosphate group, and locations of higher energy chemical bonds. The hydrolysis of ATP breaks the terminal phosphate bond, releases energy, and leaves ADP, an inorganic phosphate, and hydrogen ion. The hydrolysis of ATP breaks the terminal phosphate bond, releases energy, and leaves the AMP, inorganic phosphate, and hydrogen. So let's go over the biological energy system. So there are three basic energy systems that include in muscle cells to replenish ATP. Those systems include the phosphagen system, glycolysis, oxidative system. So let's start with the phosphagen system. So the phosphagen system provides ATP primarily for short-term high-intensity activities and is an active start to all exercise regardless of the intensity. Creatine kinase catalyzes the synthesis of ATP from CP and ADP. As well as in the phosphagen system, there are ATP stores. The body does not store enough ATP for exercise, so some ATP is needed for basic cellular function. And what the phosphagen system does is that it uses the creatine kinase reaction to maintain the concentration of ATP, and the phosphagen system replenishes ATP rapidly. Also in the phosphagen system is the control, which is done through the law of mass action. The concentration of reactants or products or both in solution that will drive the direction of the reactions. As we said, glycolysis is another biological energy system, and glycolysis is the breakdown of carbohydrates, either glycogen stored in the muscle or glucose delivered in the blood to resynthesize the ATP. And so the next slide is gonna show glycolysis. Okay, and so as you see, the end result of glycolysis is pyruvate may proceed in one of the two directions. Is that pyruvate can be converted to lactate. So an ATP resynthesis occurs at a faster rate but is limited in duration, and the process is sometimes called the anaerobic glycolysis or just fast glycolysis. And two, pyruvate can be shuttled into mitochondria, and is when pyruvate is shuttled into the mitochondria to undergo the Krebs cycle. The ATP resynthesis rate is slower, but it can occur for a longer duration if the exercise intensity is low enough. And this process is often referred to as aerobic glycolysis or slow glycolysis since the rate is slower. And so in glycolysis, you also have the formation of lactate, as we had said. And the formation of lactate from pyruvate is catalyzed by the enzyme lactate dehydrogenase. The end result is not lactic acid, and lactate is not the cause of fatigue or any cramps or, as I would say, like soreness. And it's going to be glucose plus 2 inorganic phosphate, 2 ADP, and that will yield 2 lactate plus 2 ADP and H2O. Now you now have the Cori cycle, and so what this is that lactate can be transported in the blood to the liver where it is converted to glucose, and this is what the cycle is referred to as. And continued in glycolysis for the biological energy system is the Krebs cycle, and it's the pyruvate that enters the mitochondria is converted into acetyl-CoA, and acetyl-CoA can enter the Krebs cycle, and the NADH molecules that enter the electron transport system, they can also be used to resynthesize ATP. This is where you have glucose plus 2 inorganic phosphate plus 2 ADP plus 2 NAD, which will yield the 2 pyruvate, the 2 ATP, the 2 NAHD, and now 2 H2O. And now continuing with glycosis, you have the energy yield of it. And glycolysis from one molecule of blood glucose yields a net of 2 ATP molecules. So for every blood glucose, you have 2 ATP molecules. And glycolysis from muscle glycogen yields a net of 3 ATP molecules. So it's one more. The controlled glycolysis is stimulated by hot concentrations of ADP, anorganic phosphate, and ammonia, and by a slight decrease in pH and AMP. And it is inhibited by markedly lower pH, ATP, CP, citrate, and free fatty acids. And is also affected by hexokinase, phosphofructokinase, and pyruvate kinase. And continuing glycolysis, lactate threshold on the onset of blood lactate, so lactate threshold, also shown as LT, represents an increasing reliance on anaerobic mechanisms. So LT is often used as a marker of anaerobic threshold. Now, and what is the lactate threshold? Well, the lactate threshold is the exercise intensity or relative intensity at which blood lactate begins an abrupt increase above the baseline concentration. So in the next figure, you would see how lactate threshold and onset of blood lactate accumulation, otherwise known as OBLA. And so lactate threshold and onset of blood lactate, so LT begins at 50% to 60% of maximal oxygen uptake in untrained individuals, and in aerobically trained athletes is from 70 to 80%. So OBLA is the second increase in the rate of lactate accumulation, and this occurs at a higher relative intensities of exercise. And it occurs when the concentration of blood lactate reaches 4 millimoles per liter. So the oxidative system now of the biological energy system is where the primary source of ATP is at rest and during the low intensity activities. So it uses primarily carbohydrates and fats such as substrates. And so for glucose and glycogen oxidation, the metabolism of blood glucose and muscle glycogen begins with glycolysis and leads to the Krebs cycle. NADH and FDH molecules transport hydrogen atoms to electron transport chain. This is where ATP is produced from ADP. And so in the next slide, you'll see the Krebs cycle. And now in the next slide, you'll see the electron transport chain. So now we have fat oxidation. So fat oxidation is where triglycerides stored in fat cells can be broken down by a hormone-sensitive lipase. This releases free fatty acids from the fat cells into the blood where they can circulate and enter muscle fibers. So in fat oxidation, there are some free fatty acids that come from intramuscular sources. And free fatty acids enter the mitochondria, are broken down, and form acetyl-CoA and hydrogen protons. And the acetyl-CoA enters the Krebs cycle, and the hydrogen atoms are carried by NADH and FADH to the electron transport chain. So now in protein oxidation, protein is not a significant source of energy for most activities. Protein is actually broken down into amino acids, and the amino acids are converted into glucose, pyruvate, or various Krebs cycle intermediates to produce the ATP. And so the control of the oxidative or aerobic system is that the isocitrate dehydrogenase is stimulated by ADP and inhibited by ATP. The rate of the Krebs cycle is reduced by NAD and FAD and are not available in sufficient quantities to accept hydrogen. The ETC is stimulated by ADP and is inhibited by ATP. And in the next slide, you see the metabolism of fat, carbohydrate, and protein. And so now these are some key points. In general, there is an inverse relationship between given energy system maximum rate of ATP production and total amount of ATP it is capable of producing over time. Another key point is that the extent in which of the three energy systems contributes to ATP production depends primarily on the intensity of muscular activity and secondarily on the duration. At no time during either exercise or rest does any single energy system provide the complete supply of energy. And so now you have substrate depletion and repletion. So you have phosphagens. Creatine phosphate can decrease markedly, 50 to 70% during the first stage, which is 5 to 30 seconds of high intensity exercise, and can be almost eliminated as a result of very intense exercise due to exhaustion. So in phosphagens, the post-exercise phosphagen repletion can occur in a relatively short period. Complete resynthesis of ATP appears to occur within 3 to 5 minutes, and complete creatine phosphate resynthesis can occur within 8 minutes. So now for glycogen, the rate of glycogen depletion is related to exercise intensity. So at relative intensities of exercise above 60% of maximal oxygen uptake, muscle glycogen becomes an increasingly important energy substrate. The entire glycogen content of some muscle cells can become depleted during exercise. And so for glycogen, you have the repetition of muscle glycogen during recovery is related to post-exercise carbohydrate ingestion. And so repetition appears to be optimal if 0.7 to 3.0 grams of carbohydrates per kilogram of body weight is ingested every 2 hours following the exercise. And for the next slide, you will see the low intensity steady state exercise metabolism. And so the key term for this is the EPOC, which is the excess post-exercise oxygen consumption. Oxygen uptake above resting values used to restore the body to the pre-exercise condition, also called post-exercise oxygen uptake, oxygen depth, or recovery O2. And now in the next slide, you'll see the high intensity non-steady state of exercise metabolism. And so you'll see here that it's 80% of maximum power output. The required VO2 here is the oxygen uptake that would be required to sustain the exercise if such an uptake were possible to attain, because it is not possible that the oxygen deficit lasts for the duration of the exercise. And so the key point is this, is the use of appropriate exercise intensities and rest intervals allows for the selection of specific energy systems during training and results in more efficient and productive regimens for specific athletic events with various metabolic demands. And so there are different metabolic specificity of training. So you have interval training, which emphasizes bioenergetic adaptations for a more efficient energy transfer with the metabolic pathways by using predetermined intervals of exercise and rest period. There's much more training that can be accomplished at higher intensities, and it's difficult to establish definitive guidelines for choosing specific work to rest ratios. You also have high intensity interval training, otherwise known as HIIT, brief intervals bouts of high intensity exercise with intermittent recovery periods to elicit the cardiopulmonary, metabolic, and neuromuscular adaptations. And in HIIT, their cumulative duration of intensity of active portions should adequate to several minutes above 90% of VO2 max. In HIIT, suggested work to rest ratios are greater than 1:1, and when used in conjunction with other training sessions, it may result in greater stress and risk of energy. And last but not least, you have combination training. Combination training adds aerobic endurance training to the training of anaerobic athletes in order to enhance the recovery, and because recovery relies primarily on the the aerobic mechanisms, and they may produce anaerobic performance capabilities, particularly high strength, high power performance. And for combination training, it can reduce the gain in muscle girth, maximum strength and speed, and power-related performance, and it'd be counterproductive in most strength and power sports to do combination training. And this is the summary for Chapter 3. Thank you.